Hey guys. Okay, today we're going to talk about chapter 6 from your OpenStax book that is dealing with acellular pathogens, including viruses. So we'll spend most of our time on viruses, and then we'll talk at the end a little bit about some other acellular pathogens, meaning anything besides bacteria or protists or other other things that we've talked about in the previous two lectures that Professor Marovich provided for you guys when we talked about prokaryotic diversity, then we talked about eukaryotic diversity. Well, now we're going to talk about acellular pathogens. Okay, so we're going to round out this section of this unit. Okay, so that you're, I'll talk to you more about what's coming up on your exam another time. Okay. Okay, so this is from your OpenStax book, you guys. This is just showing you in 2014, there was a pretty serious Ebola virus outbreak, excuse me, in Western Africa. And this map is just showing you the different countries in that region. It's shown to you here where we're looking at here. And it's showing you all the different cases and deaths that were caused by that particular virus. And so now this is how we kind of epidemiologists sort of relay information to us. They usually show us a map, and then they'll tell us how many cases there are and how many deaths. So cases will tell you how widespread it is, and deaths will tell you how pathogenic it is, right? So we started to do this, and we still continue to do this with COVID as well. It's very similar. And so such epidemics or pandemics like COVID are now widely reported and documented. But viral epidemics are sure to have plagued human populations since the origin of our species. So these things have been around since we have and before. We're just getting better and better at documenting that and figuring out where it came from. So some of you have started listening to the Patient Zero podcast, and they're talking a lot about that as well, trying to figure out what the causative agent of this mysterious sort of juvenile arthritis symptoms were, or how to figure out what caused that, or what the causative agent was of those things. Turns out it's Borrelia burgdorferi, and now we call it Lyme disease. So same idea, and so that whole podcast talks about how that was discovered and how difficult it can be to figure out one, what causes an infection or a disease, and two, how to stop it, how to contain it, how to treat it, right? And so there's just a lot of pathogens that where they'll have this kind of data here that epidemiologists will put out for us. Okay, so we're going to focus mostly on viruses then for this lecture. Okay, so one thing that's very unique about viruses is that they fall into the category of obligatory intracellular parasites. And that just means that parasite means they live in or on us, a host. They're intracellular, which means they have to get not only inside a host, but inside a host cell. And they have to do that to replicate. They can't live outside of a cell to survive. Okay, so they're obligate, means that they have to do this. Intracellular means they have to get inside the cell. And the parasites, which just means something that causes damage, that lives in or on us. Okay, so they contain either DNA or RNA, but never both. So they're among some of the only entities on the planet that we know of that can exist with just RNA. They don't have to have DNA. So everything we've talked about so far, you guys, all the bacteria and the eukaryotic pathogens that we've talked about in the previous two lectures, all those cells contain DNA. But now we're going to talk about things like viruses and other sort of acellular pathogens that only have RNA. Okay, so very unusual. And in fact, there's one that I'm going to talk to you about that doesn't contain either. Okay, so really interesting little pathogen there. Okay, that'll be at the end of the class today. Okay, so what do viruses have then? Like I said, they have DNA or RNA, and that DNA or RNA is always contained in a protein coat. Okay, so they have a sort of capsid that's made of protein that surrounds the DNA or RNA. So they all have to contain at least those two things, either RNA or DNA and a protein coat. Sometimes some viruses contain an extra layer called and it's a phospholipid envelope. So it's kind of like a membrane that surrounds the protein coat, and that protein coat surrounds the genomic DNA or RNA. Okay, but these are just half of the viruses or some portion of the viruses, not all of them. Okay, they don't contain any ribosomes or any other organelles. They don't have any way to generate ATP, so they have to pirate that from the host. Okay, and they're really small. They're like 1/10 to 1/100th of the size of bacterial cells. So we've already seen how small bacterial cells are in our labs the last couple of weeks, right? Because it's really hard to see them unless you're looking with your oil immersion lens. That's 1,000 times magnified. So these guys are another 10 or 100-fold smaller than that. So they can't be seen with, typically can't be seen with the light microscope that we use in class. Okay. So let me show you some comparisons between viruses over here on the right and typical bacteria. Now there are rickettsia and chlamydias. Professor Marowich told you guys a little bit about rickettsia and chlamydia in her lectures that I posted for last week with some of the prokaryotic diversity. Okay, so these guys kind of fall in the middle of viruses and bacteria. They're considered bacteria, because you can see here they fall into this category, but you'll see that they have some resemblance to viruses, right? But I mostly want you guys just to see this column versus this column. Okay, so typical bacteria. Are they intracellular parasites? Typically no, but rickettsia are, and chlamydia, and viruses for sure. They have to be intracellular. Bacteria contain a plasma membrane, viruses do not. They replicate by binary fission, viruses do not. We'll talk about how they replicate today. Can they pass through typical filters, bacteriological filters? No, that's why they're called that, so we can stop the flow of bacteria through a filter. But viruses are so small that they can get through those typical pores in a filter, okay? And you can see here, some of these guys can, some of these guys can't. Do they possess both DNA and RNA? Yes, bacteria do. They contain DNA and several types of RNA, but viruses don't. They contain one or the other. Can they make ATP? Yes, typical bacteria, no, viruses. Have ribosomes? Yes, for bacteria, no for viruses. Are they sensitive to antibiotics? Yes, for bacteria, no for viruses. Viruses are not susceptible to any antibiotic. All right, so never ever get an antibiotic unless you know you have a bacterial infection, because it doesn't do any good. Sensitive to interferon. Interferon is a kind of treatment that is effective against viruses, but not bacteria. Okay, so this just gives you a nice sort of summary of what some characteristics of viruses versus bacteria. And then we sort of have this wacky group of bacteria in between here, you guys, that have some things in common with viruses, but some things in common with bacteria. So they're interesting little buggers. Okay. Okay, so the spectrum of host cells a virus can infect. All right. What did I want to say here about that? Maybe I didn't finish my statement here. So it's pretty small, right? So most viruses infect only a specific type of cell and in one host. Okay, sometimes they can infect more than one host. Like we know influenza can infect pigs, can infect birds, can infect humans, but it's typically the same host cell in that case. Okay, but most viruses infect only specific types of cells in one host. So they're very host specific. Okay, very host specific. So the spectrum of host cells a virus can infect, that should say, is very narrow. Okay, that should say is very narrow. I can let me see if I can fix that. Yep. Okay. Okay, and why is that the case? It's because that the virus and the host cell have a specific interaction that has to occur. So there's a specific host attachment site on the cell surface of the host and other cellular factors that determine what cell the virus can attach to. Okay, so it's very specific to the external part of the cell that allows a virus to attach. And so not every cell is going to have it, and certainly not every species is going to have it. Okay, so it's a very specific interaction between the host cell and the virus. Okay, it's a specific interaction. Okay, we talked about how small they were. Remember, bacteria, you guys, are anywhere between 1 and 5 or 10 micrometers long. Micrometers. Viruses are anywhere from 20 to 1,000 nanometers. Nanometers, you guys. So that's anywhere between 0.02 and 1 micrometer. Okay, so a nanometer is 1/1000th of a micrometer. Make sense? So these are way smaller. Okay, we're using 1,000-fold smaller of a unit here when we talk about nanometers compared to micrometers. So they're much smaller typically. Okay, bacteriophages are a specific type of virus, you guys. I've talked to you about this before. These are viruses that infect bacteria. Okay, so every single cell known has a virus that will infect it, including bacterial cells. Okay, so bacteriophages just get their own name. Okay, phage refers to eating. And so we used to think that these things were eating bacteria cells, and I'll show you a picture of why. And so we just call this bacteriophage because it looks like they're eating the bacteria cells. Okay, so there's a whole new, not new, but it's kind of made news again lately of using bacteriophage to treat bacterial infections as opposed to antibiotics. Right, so if you're interested in something like that, you should look into that. I'll probably talk about it later in the semester, but antibiotics are known to, if we overuse them, it will result in resistance to those antibiotics. And so it's this race between us and the bacteria. We're trying to develop new antibiotics or discover new antibiotics, but the bacteria are mutating faster than we can do that. And they're becoming more and more resistant to the known antibiotics that we have. And so phage therapy is a new kind of science where we use a specific bacterial virus, a bacteriophage, that will specifically infect or specifically attack a bacterial infection that we have. It won't affect our cells because it's specific only to that bacteria. So some people are, feel kind of squeamish about that. Well, how do you treat your bacterial infection? Well, let me drink a cocktail of viruses, right? So it sounds like something you might not want to do, but actually it's much more specific and it's much more effective. And so there's lots of scientists working on that at the moment. Okay. Okay, so here's a typical bacteriophage, you guys. They seriously look like this, these crazy looking alien looking structures. Here's an actual picture, an electron micrograph of a bacteriophage. Here's the cell it's infecting here. Here's the membrane. And it actually will sit down and dock onto this membrane, and it will inject its genome, whether it's DNA or RNA, through this sheath here down into the cell. So it actually looks like a syringe. Right, so these guys are pretty weird, but that's exactly how they work. And the rest of it will stay on the outside. And then once the DNA or RNA is inside the host cell, it just starts taking over. It'll start expressing its RNA and its proteins, and those proteins will take over the cell machinery, and it will start reproducing many, many, many copies of itself until it explodes the host cell. And many of these bacteriophages will go and infect new cells. All right, so they're pretty efficient at what they do. This just shows you another example of the size of these guys. So here, for example, is a typical bacterial cell, right? So it's probably maybe that one's probably like 10 micrometers if we put it from here to here, right? So maybe about 8 to 10 micrometers long. But here's, a typical, let's see, here's a flu virus. The flu virus is actually kind of big, right? But it's on the order of 10 nanometers as opposed to 1 micrometer, right? Smallpox is here. Here's the polio virus down here at around 20 or 30 nanometers. Okay, so much smaller than typical bacteria. Here's a red blood cell. So that's a little bit bigger than a bacterial cell. Plant cell, animal cells are up here around 50 micrometers. Human eggs, pollen, frog eggs are a little bit bigger. Okay, just to give you an idea. Okay, here's another example of that from a different textbook, but you get the idea. So here's a red blood cell. Here's E. coli. Here's chlamydia. Remember I was telling you how chlamydia kind of falls between viruses and bacteria? Well, sure, it certainly does in size as well. Here's the Ebola virus. Here's a bacteriophage. Okay, here's some other examples here as well. Okay, so it just shows these are mostly all viruses in here. Okay, there's some other things in here that we'll talk about later that aren't viruses, but you can see how much smaller they are compared to the typical E. coli bacterial cell. Okay, and this is just another example that shows you of the filter size I was talking about. And so if you have this setup like this, where you want to sterilize your media, right? And you want to push unsterile media into this flask, you want to have a filter here that has pores in it that will block whatever you don't want to get into your media. So in this case, you have a filter that's got the size of 5 micrometers. And let's see what we're trying to do here. So in this case, you're blocking out things, anything that's bigger than 5 micrometers here. So let's say that these red guys are E. coli, right? So when you push that through, none of those guys get through. So you do prevent E. coli from getting through. But the smaller things, the viruses, let's say viruses are the blue and the green guys, okay, they're actually going to get through. So if you make the pore size smaller, so here it's 0.2 micrometers instead of 5, so much, much smaller. Now you're going to block the blue viruses, but it's still not small enough to block whatever these green guys are. Okay, so in this case, the viruses, are green, bacteria are blue, and animal cells are red. Okay, so let's go back. So here you have a 5 micrometer pore size, so viruses and bacteria can get through. If you shrink that to 0.2 micrometers, now you've stopped bacteria and the animal cells from getting through, but you still let the viruses get through. So you still have to make that even smaller. So it's hard to make filters small enough so that viruses don't get through. Okay, that's kind of the whole argument about mask wearing, right? Well, virus particles are small enough to get through the fibers in the mask. That's probably true, but the viruses that cause COVID don't move by themselves. They're always attached to some kind of respiratory droplet or aerosol. And so that's much bigger than the pore sizes in our mask. And so it's the droplets that get stopped, and the viruses are almost always attached to those droplets. Okay, so together, they're too big to get through the pore size. Okay, so that's why masks are effective. Okay, so let's get into some viral structure then. We talked about size, let's talk about their structure. So they're pretty simple, as I've already said, right? So we call a complete, fully developed viral particles a virion. Okay, a virion. So a virion consists of at least 2 things. 1, nucleic acid, and that can be either DNA or RNA. And that DNA or RNA can be either single or double-stranded. Okay, so the DNA can be single-stranded or double-stranded. The RNA can be single-stranded or double-stranded. And that that nucleic acid can be either linear or circular. So all combinations of DNA or RNA, single-stranded, double-stranded, DNA, RNA, circular, linear, all right? So they're not picky, right? They have there's all kinds of viruses that exist. And then that nucleic acid must be surrounded or protected by an outer coating called a capsid, all right? So it's a protein coat made of subunits called capsomeres. All right, so there's little subunits in the capsid that are called capsomeres. So a virion must consist of those 2 things, okay, to be considered a full, complete viral particle. All right, so those 2 things, nucleic acid and a capsid. Some of them have an extra outer coating called an envelope. An envelope looks just like a lipid bilayer, right? It's got, like I said, a lipid bilayer, it's got proteins stuck in there, it's got carbohydrates stuck in there, just like a plasma membrane would look. Okay, that's what it looks like. And then sticking off of that envelope are projections that come out like receptors or antennae that come out of that outer surface. And those are the things that allow it to have a specific attachment or specific affinity for a specific host cell. Okay, it's those spike proteins or those spike molecules that give it its specificity. So those spikes are typically the ones that will attach to some human cell receptor and makes that a very specific interaction. Okay, the capsid has it too. The capsid has to have some specificity as well, because if you don't have an envelope, you still have to be able to attach to a host cell. So the capsid does have some of that as well, that specific outer molecules that it will allow attachment to a host cell. And we'll talk about how each of these gets inside a host cell in a minute. Okay, so here's some examples of both types of those viruses. So this is one that does not have an envelope. Okay, and so you can see the capsid here is all these yellow sort of structures here, and those are the capsomeres. So each one of these little yellow ovals is a capsomere, and all of the yellow together is called the capsid. And this one happens to have DNA that, or a nucleic acid that's DNA. All right, so that's here. And you can see on that capsid are some spikes made of glycoproteins. Okay, so glycoproteins means part carbohydrate, part protein. Okay, so the spikes don't only exist on envelopes. So I didn't mean to imply that back here. Okay, so spikes, I shouldn't, this doesn't mean that they're just envelope viruses. Spikes can occur whether it's enveloped or not. Okay, makes sense? Okay, so here's one that's not enveloped again. So the capsid is here in yellow, and the individual ovals are the capsomeres. These are spikes, okay, made of glycoproteins, and inside is the nucleic acid, in this case, DNA. Okay, so here it is attached to the host cell. Okay, and the other type was the enveloped virus. In this case, this is the HIV retrovirus. It causes AIDS. And so here you can see this structure right here is the capsid. And then inside you see individual strands of RNA. This is an RNA virus, okay, inside the capsid. But then surrounding that is another layer called the envelope. Okay, so this is a membrane here. And then these big purple guys are spikes that are made of glycoproteins. Okay, so whether you're enveloped or not, you have these external spikes, and that's what's going to allow for attachment to the host cell most of the time. Okay, so here's an example. Here's showing HIV attaching to its host cell in this case. Okay, makes sense? So spikes either way. Here you can see single-stranded RNA. Here you can see DNA. Here you can see that the capsid is not surrounded by an envelope, but here you can. Okay. Okay, so what other differences can they have? Well, the capsid can be have various structures. So you can have helical virus, which means that the capsid is hollow and cylindrical. You can have polyhedral shaped viruses, which means the capsid is many-sided. I'll show you that. Again, you can have envelope viruses, which I already talked to you about, or you can have what they just throw everything else into what's called complex viruses, which means they have a really complicated, unique structure and don't really fall into any other typical categories. So let me show you each kind. Okay, so here's a helical virus shown to you here. So you can see the nucleic acid in red in here, and then the capsid is the yellow that's forming this helix around that nucleic acid. Okay, so that's helical. Here's the polyhedron. In this case, it's called icosahedral, and it shows to you it has many sides. Okay, so it's got like facets, like a diamond. All right, so that's called polyhedral, meaning many sides. Okay, so you can kind of see that there. Here you can see an actual, it's kind of more of an artist rendition of the virus, but you can see it's got many faces, corners, okay, but it's got a very geometric looking shape. Okay, and that's the capsid. Inside here, you can see the nucleic acid. And then here's an example of a complex virus that doesn't really fall into any category. It just means complex. So here you have an envelope out here. The capsid is here, right? And then the nucleic acid is the blue squiggly inside here. Okay, so many of them fall into here. Bacteriophage also fall into this category. Okay, I'll show you that. Here's another example of a non-envelope polyhedral virus. So inside in purple is the nucleic acid. Outside is the capsid, right? These blue balls, and each one of these spheres or balls is a capsomere. Okay, but you can see how it's got a very specific geometric shape here, polyhedral shape. Okay, but no envelope. So there's an example. Here's an envelope virus, and it's helical in shape, the capsid. So the capsid is in blue, and it's going to be helical inside, and then the nucleic acid is the purple, and then the envelope is this gold or yellow colored outer structure here, and it's got these green spike glycoproteins that are sticking off of the envelope. Okay, so enveloped, helical virus. Okay, and then we can also categorize it by whether it's DNA or RNA, whether it's linear or circular, or if it's double-stranded or single-stranded. Okay, so there's lots of ways to categorize viruses. Here's a non-enveloped helical virus. Okay, just to show you another example. So you can see here how the capsid is going to form a helix around the DNA or RNA that's inside, right? This is actually the Ebola virus. Ebola virus has a very characteristic shape like this. Okay. Here is a complex virus, another one. This is the bacteriophage I already showed you. So it's got this head region here, which is the capsid, and then it's got this sheath region here, which has other proteins surrounding it. And this is kind of what is the syringe. So the DNA will get just literally injected through this sheath down into the host cell here. Okay, so you can see that happening here. Okay, here's another example of a complex virus here, orthopox virus. Okay, just shows you another example. All right, so I'm not going to ask specifics if you know exactly what virus family falls into these categories, but if I show you an example of a virus, a picture of a virus, you should be able to tell me, is it helical? Is it polyhedral? Is it complex? And why? Okay. Okay, and viruses aren't named like other species. There's no genus and species name like there is for bacteria that we've talked about. So when we say, Micrococcus luteus or Escherichia coli or Bacillus megaterium, they don't work like that because they're not living. I don't know, maybe whoever decided to name them decided they weren't going to follow convention, right? They're not considered living because they aren't made up of a cell. Remember the first part of the cell theory says that all living things must be made up of one cell, right? And they're not. So they're not considered living. So none of the stuff that we're talking about today is considered living because they're all acellular pathogens, okay? But there is a way to name them, and I'm not going to go into a lot of detail here, but just a few things here. So the genus names end in virus. Okay, so I guess we do sort of, do talk about genus and species, but it's not typical. It's not the same. We don't name them the same way. Okay, so they always end in virus. The family name ends in Viroidae. Okay, so Viroidae. I'll show you some examples. And then if there's an order, it ends in ALES. Okay, so remember we talked about order, family, genus, species. Okay, so but typically when we talk about viruses, you guys, we only really talk about whatever it is virus, like coronavirus or influenza virus or Ebola virus, right? And so we really don't use the fancy names very often when we talk about viruses. Okay, it's just not convention. But there are species of viruses. We'll call them that. So a species of virus is very similar to a species of any other living organism. It's a group of viruses sharing the same genetic information and ecological niche, or in this case, host. Remember, because viruses have to have a host. So they're sharing the same genetic information and the same place they live, their host. Descriptive common names are used for species typically, and subspecies are designated by a number. So when we talk about E. coli, for example, okay, so E. coli would be the species name. Escherichia, that's a bacteria, sorry. Influenza, okay, so influenza would be the common name, and then we might say influenza H1N1, right? So that would be designated by a number. So there's a subspecies of influenza that can be designated according to some proteins on the outside of its envelope, the H protein and the N protein. And so different versions of those have different numbers. And in that case, the number was 1. So that's an example of a subspecies of influenza. Okay, so how do we grow them in the lab? Before we go into how they replicate, you guys, I want to talk about how you grow them in the lab. So they're definitely not as easy to grow in the lab, you guys, as bacteria are, because bacteria can grow outside of a host cell, right on a plate. You guys have done it already in lab, right? So they're very easy to grow. Most bacteria are easy to grow outside of a lab. But viruses can't grow outside of a living cell. And so we have to have some cell also to grow so that we can have our viruses grow. So it's tricky. It's much trickier to grow viruses. So bacteriophage, again, are those viruses, you guys, that grow inside bacteria. So bacteriophage are a little bit easier to study because it's very easy to grow bacteria, as you know, right? And so if we grow some bacteria, infect it with bacteriophage, and let those both grow up together on a plate, on an agar plate, bacteriophage forms something called plaques in that lawn of bacteria. And plaques are clearings on a lawn of bacteria on the surface of the agar because they have eaten through the bacteria. Okay, so plaques are just clear zones on a lawn of bacteria on an agar plate. Okay, so I'll show you a picture of that. So each plaque is basically corresponds to one single virus, and it can be expressed as a plaque forming unit. So you know we've talked about bacterial colonies, and sometimes they're called CFUs or colony forming units. That's because they all came from one bacterial cell to start with. And so we call them colony forming units. Those cells are called colony forming units. Well, plaques also came from one viral particle, one bacteriophage to start with. And then that grew and grew and grew, and it created this clearing zone on the plate. And so those plaques, because they came from one initial bacteriophage, we call that phage a plaque forming unit. Okay, so it means the same thing as just plaque. It just reminds us that, oh yeah, each plaque, even though there were millions of viruses that created that plaque, only one started the whole thing. Just like one bacteria cell starts a colony. Okay, so let me show you a picture of that. Don't look at this picture yet. This is referring to something on the next slide, but this is what a plate of bacteria look like when they've been infected with a bacteriophage. So look here. This whole plate is covered with a lawn of bacteria. You can't really tell on this slide, but the whole thing is covered. And if you infect it with a phage or mix it with a phage that infects it and you let it grow, you'll see the phage will start to kill the bacteria and create these zones of no growth called a plaque. And so these plaques can be counted, just like you guys are doing in lab this week, where you're going to count colonies. In this case, you're going to count phage or plaques. So it tells you how many phage were in that experiment. And this is just a dilution plate. Just like you guys dilute your bacteria, here you dilute your phage. So here you have too many to count. Here is probably just the right number. We want between 30 and 300, just like in lab. Here's too few to count, right? You can see here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So too few to count because we want between 30 and 300, just like with the bacteria. Makes sense? So this is the one that you'd want to count. And that would tell you, when you do a little bit of math, how many phage you started with. Okay? But I just want to point out what that looks like. So back here, whoops. Okay, so back here, we're talking about bacteriophage formed plaques, which are clearings on a lawn of bacteria on the surface of the agar, because those phage are viruses that have infected the bacteria in that lawn and have systematically killed all the bacteria in that plaque. And so that's a clearing on the plate. Okay, makes sense? Okay, here's another example to show you a little bit closer. So here's the lawn of bacteria all on this plate, and you can see these clearings here of the where the plaque has infected the bacterial cell. So what about in other kinds of cells, not just bacteria? What about other kinds of cells? So viruses infect bacteria, plants, animals, fungi, protozoa, all kinds of cells. So we could talk about all of that, but we're really just going to focus on animal cells because that's what we are. And that's what we're interested in. That's mostly what we're interested in when we think about viruses, right? How they infect humans and other mammals too. Okay. But bacteriophage are interesting because you guys work with bacteria too. And so I like to point that out. Okay, so how do we grow animal viruses? Well, there's two choices. One, in actual living animals, which is a way that many virologists study viruses, but this can be expensive. There can be some ethical issues involved. Not everybody likes to work with live animals. Infecting live animals with viruses is not a very, fun, nice thing to do. So, it's not for everybody. Let's put it that way, right? So many other people will study viruses in like embryonated eggs. So in chick eggs, for example, that have an embryo growing in it. Okay, so the virus gets injected into the egg, and then viral growth is signaled by changes or death of the embryo. So then you know the virus has grown. Okay, so we have to have a way to propagate viruses to study them, right? And so we either have to do it in the living animal itself or in an egg of that animal, okay? Because we have to have a way to grow the viruses to be able to study them. Right, so here's an example of a scientist that's injecting a virus into a chick egg here, okay? So the cells within chick eggs are used to culture different types of viruses. And so this just shows you where in the egg you can inject various viruses, okay? So they can be replicated in various locations. There's all these membranes in here, right? It can be in the amniotic cavity. It could be in the chorioallantoic membrane, right? It could be in the yolk itself, right? So it just depends on where your virus replicates best, where you'd want to inject that virus into this chick, okay? Okay, so let's go into the viral life cycle then, and how the virus replicates. That's going to be the majority of our talk today, okay? So for a virus to multiply, it needs three things. It must invade a host cell. Remember I said it's intracellular. It must take over the host's metabolic machinery. So it pirates the machinery from the host and takes it over, and it typically will kill the host cell when it does so. And then it must escape to find a new cell to infect. Okay, so it has to get in, it has to take over the host machinery and make more of itself, and then it has to be able to escape and find a new cell to infect. That's what a successful virus does. So really, a virus that kills its host is not really the most successful virus, because then it's a dead end, right? If it kills its host, then it's not likely going to be able to spread to a new host. And so a good virus, a successful virus, is one that makes you sick, but not too sick, because the virus needs you to be moving around so that you can come into contact with other susceptible hosts. So viruses like the flu and the cold are pretty good viruses, because typically we don't stay away from people like we should when we have those illnesses, right? Ones that cause STDs that don't make you sick or sick enough, right, are good viruses from their point of view, because it means you're still healthy enough to pass that virus to someone else. Okay, so that's a good virus. Is one that doesn't make you too sick, so that you can easily pass that virus on to a new healthy cell. All right. So two major categories we're going to discuss, two major viruses again: bacteriophage, viruses that infect bacteria, and then animal viruses. So just kind of the theme for today is what we're going to continue on with, those two types of viruses. Okay, so first the bacteriophage. So bacteriophage can go through two different kinds of cycles. The first is called a lytic cycle, and the second is called a lysogenic cycle. So two different phases there. The lytic cycle is sort of the fast and furious: get in, make more of yourself, burst the host cell, get out, find a new cell to replicate or to kill, to get into, right? So it causes lysis and death of the host cell, moves on to a new host cell, and repeat. Okay, so that's the lytic cycle. So lytic or lysis means to kill or to destroy the cell. So that's common for bacteriophage to do that. The lysogenic cycle is a little bit slower, and it doesn't have to be, well, it's a little bit slower because the phage DNA doesn't just get in there, make more of itself and get out. It actually will incorporate into the host chromosome. So it doesn't replicate, it will hide. It will just incorporate, recombine into the host DNA. And it may just stay like that for a long period of time. Okay, staying dormant until maybe the conditions are better that it can replicate. So the virus kind of knows when to do this. It knows when it should just hide, and it knows when it should pop back out and start the lytic cycle again. So it can kind of go back and forth between these two. Okay, not every virus can do the lysogenic cycle, right? Everybody can undergo the lytic cycle, but for bacteriophage, only some of them can undergo the lysogenic cycle. All right. So when this happens, you guys, when a lysogenic cycle happens, sometimes it can cause what's called phage conversion, which means that when this phage DNA incorporates into the host DNA, it changes the DNA of the host. Depends on where it incorporates. It might knock out a gene. If it incorporates into an existing gene, then maybe it stops that gene from being expressed. And so you can convert that host bacterial cell into something different, okay, depending on where the viral DNA inserts into the chromosome. That makes sense? And so that's called phage conversion. Sometimes that will kill the host cell. Sometimes it'll just change the host cell. And sometimes it'll make that host cell better fit for its environment. Sometimes it'll make it less fit for its environment. Either way, or anything, anyway, it's still called phage conversion. Okay, makes sense? So it has to do with that phage DNA incorporating into the host DNA and maybe causing that, changing that host DNA to something else. So it makes the host different. Okay, better, worse, dead, right? It just depends on where it actually inserts. Okay, understand the difference? This is fast and furious, never inserts into the host chromosome, just gets in there, starts replicating, makes more copies of itself, and then busts out of that cell and goes find someone else, go to find someone else. This is a little slower. It's sort of deciding whether or not it should stay dormant for a while or if it should pop out and start replicating again. Okay. Okay, so here's the lytic cycle of a specific kind of bacteriophage called T-even, or maybe T2 or T4. Even just means the number. Okay, so 4, 8. Okay, those are called T-even, and they have a lot in common. Okay, so this is a lytic cycle. So there are several stages that must happen. I'll list them out first, and then I'll show you a figure after this slide. So the first thing that has to happen with any virus, including bacteriophage, is that it must attach by the tail fibers to the host cell. So there has to be some specific attachment place where the tail fibers recognize the bacterial cell that it's specific to, and they'll be able to create a bond. So here's the tail fiber, here's the receptor on the host cell, and it will be able to bond there, specific bond. Okay, so that's called attachment. Then, once it's attached, the phage lysozyme, so the phage produces lysozyme, it opens up the bacterial cell wall. The tail sheath contracts to force the tail core and the DNA into the cell. So it basically sits down on the cell like this, and then it contracts to just allow it to sit down onto the cell, right? The tail fibers or the tail sheath contract, and then the DNA can go into the cell. Okay, that's pretty cool. Pretty cool little mechanism those bacteriophage have. Okay, so that's called penetration. When the DNA enters the host cell, that's called penetration. Okay, then once that DNA is inside the host cell, then biosynthesis occurs. And that means biosynthesis of both the DNA and the viral proteins. So that DNA is now going to start to express, its genes, transcribe and translate its genes, and make new DNA and also make new proteins. So it's going to make new phage particles once it's inside the host cell. But it takes all of the host enzymes to do that, right? So it's going to take the DNA polymerase. It's going to take the RNA polymerase. It's going to take the ribosomes. It's going to take the tRNAs. It's going to take the amino acids. All those things we talked about, you guys, in the genetics lecture part 1, all that stuff we talked about, the viruses will do as well, but they just steal the host enzymes to do that. All right, so that's called biosynthesis. Then they assemble all those particles together to make new phage particles. So the new DNA, the new proteins all get manufactured together, kind of like a, assembly line in a manufacturing plant, right? Where all the parts are made by somebody else, but then at some point they all get put together. Right? So that's called the maturation step. And then exit or release, right? So now the phage lysozyme breaks the cell wall again, and now all of the new phage particles release all at once, and the cell dies, the host cell dies, and all those new phage particles, you guys, go to find new susceptible bacterial cells so to be infected, to infect them. Okay, so let me show you that again. So here's the healthy bacteria cell. Here's its DNA, its chromosome. Here's the bacteriophage that has now landed on this bacterial cell. So the tail fibers here are specifically attached to some receptor on the bacterial cell wall. It's not really showing that, but it has to be, because these bacteriophage don't infect every cell. They only infect this kind of cell. So there has to be some specific interaction that happens between the tail fibers of the phage and the cell wall receptors of the bacterial cell. Okay, so that's attachment. And then once that happens, the phage will kind of sit down onto the cell wall and inject its DNA into the host. That's penetration. Once it's in there, it's going to start transcribing and translating to make new viral proteins. It's going to start replicating with DNA polymerase to make more viral genome, right? More viral DNA, this green, all this green stuff. And then eventually, that's called biosynthesis. And then it's going to assemble all of those particles together to make the mature virus. And then once it basically it just can't fit anymore inside the cell. And then the lysozyme will be produced and the cell wall will burst open and all of these new phage particles will be released. And the process starts again. Okay, so that's how plaque is formed. That's how plaque is formed. So it's one virus that sits down, and then hundreds are released. And then each of those will go infect a neighboring cell, and again, and again, and again, and eventually you end up with one of those clear plaques on a lawn of bacterial growth. Okay, makes sense? Lytic cycle. All right, notice the DNA never incorporates into the chromosome, right? It never does. That's lysogenic. Lytic means that it never incorporates and it just keeps replicating and making more and more of the bacteriophage. Okay. So another type of bacteriophage called lambda undergoes the lysogenic cycle. Okay, so this is just another kind of bacteriophage, another family of bacteriophage called lambda. The first one was called T-even phages. This is called lambdaphages. Okay, so I'll show you an example of the lysogenic cycle here. So lysogenic or lysogeny means that the phage remains latent, which means dormant. So it's not reproducing right away. It's going to just hang out, lay low, hide in the host chromosome, not making more copies of itself, okay, until conditions change and the virus now decides, okay, now is a good time for me to start replicating. Okay, so it has this extra little bit of capability to hide if the conditions aren't great for the virus to grow and spread. Okay, so it might stay latent in its host for any length of time, years sometimes. Okay, so in this case, the phage DNA incorporates host cell DNA. Okay, the phage DNA incorporates into the host cell DNA. Inserted phage DNA is known as a prophage. So if the DNA enters the host chromosome, it's now called a prophage. Okay, it's just a different term for when it's incorporated into the host cell. So it will only replicate now when the host cell replicates. So if the host cell is going to divide into two cells, it has to first replicate its DNA, right? By DNA replication. When that happens, it will also replicate the prophage DNA. All right, that's the only way that the phage can replicate while it's in this lysogenic state. Okay, only when the host cell replicates, because it's part of the host cell DNA at the moment. Okay, and again, this can result in phage conversion. That means that the host cell exhibits new properties because the phage DNA has incorporated and somehow altered the host DNA by having that new host DNA, or that new viral DNA in there, that prophage. Okay, so it just means new properties. It doesn't mean good, it doesn't mean bad. It just means new or different properties. Okay, phage conversion. So you can think of it like a mutation, but it's caused by a phage inserting, so we call it phage conversion. All right. So here's what lysogeny looks like. So this part looks the same, right? So you have assembly here, not assembly, attachment here. So the phage has sat down and attached to the cell wall of the bacteria. And then it's going to inject its DNA. That's penetration, right? But instead of biosynthesis happening at the moment, the DNA is going to incorporate temporarily into the host genome. Okay, so that's going to halt. It's going to halt that replication step. It's going to halt that biosynthesis step. Okay. So now the only way it can replicate is if the host replicates. Okay, so one cell goes to two cells, and you can see now there's two copies of the host DNA, but there's also two copies of the prophage, right? You can still see the green there. That's the viral genome. Now, under different conditions, whether it's stressful, something's going to change, and the prophage is going to pop out from the bacterial chromosome and enter the lytic cycle. So stressful meaning all kinds of things, right? It basically means a change in the environment. Okay, if that happens, then the prophage will pop out and become its own individual DNA again. And now it will enter the lytic cycle that we've already talked about. So now it will start going through the biosynthesis stage where it makes more copies of itself, starts to transcribe and translate, making more copies of the tail fibers and the head region. And then assembly will happen. And then the bacteriophage will burst out of this cell once there's so many of them. Okay, so it's the same thing, except that it might have this intermediate time period where the phage DNA has hidden or became incorporated into the host genome. Okay, so there's lots of reasons why that might happen. Not every bacteriophage can do it. Okay, but it's a way for the bacteriophage to just put the pause on, put the brakes on their replication until conditions change. Okay, so if the bacterial cell is starting to starve, then that tells the phage, uh-oh, better get out of here, right? So the stressor isn't a stressor on the virus, it's more of a stressor on the bacteria cell, so starvation or toxic chemicals. It's going to notice that the bacteria cell, its host, is not doing very well. And so then it's going to pop out and then start replicating and burst out of there before the cell dies. Okay, makes sense? So that's pretty nifty that it would know to do that, right? Like, how does it know to pop out, right? So there's all kinds of studies that look into that. Okay, so that's lysogeny. Lytic, lysogenic. Make sense? Okay, so that's bacteriophage. Let's talk about animal viruses. Okay, so we're not talking about plant viruses too much. We're not talking about fungal viruses. We're just focusing on the two types that are most commonly encountered by, in microbiology or in studying disease. Okay, so we're going to move on to animal viruses. So there's much of it is similar, you guys. The steps are similar, but because animal viruses are a little more complex, there's a few more steps involved here. So we're going to spend a little time talking about those. Okay, so the first stage, just like with bacteriophage, is attachment. The viruses have to attach to their target cell membrane. Okay, so since we're talking about animal viruses, remember, they don't have a cell wall, so they just have a membrane. So that's what the virus is going to attach to. But it's the same idea. You're going to have some spike protein on the outside of the virus that's going to attach to a specific molecule or specific receptor on the host cell. Okay, then it needs to get inside. Okay, so there's entry now. So it's a little bit different than bacteriophage because it's not injecting the DNA. It can't do that. It doesn't have that same sort of syringe feature that bacteriophage do. So there's two main ways that they do it. It's either by receptor-mediated endocytosis or it's by just fusion. Okay, so let me see if I'm going to show you those two things. Yeah, I'll show you those. So if it's a enveloped virus, the envelope is made out of the same thing that animal cell membranes are made out of. So the virus just has to fuse with that cell membrane. Make sense? Because it's made out of the same phospholipid bilayer. So it literally will just fuse with that like material and become part of the cell membrane. Okay, so that's fusion. If there's no envelope and it's just a capsid, then it goes by receptor-mediated endocytosis. So it induces the host cell to engulf the virus. So here it's sitting, okay? It will induce the host cell to open up and engulf the virus and bring it into the cell, okay? So that's called receptor-mediated endocytosis. I'll show you a picture of that here in a second too. But that's called entry, okay? Uncoating means that the capsid has to be removed from the nucleic acid of the virus, right? So with bacteriophage, it injected naked DNA. It didn't have to do any uncoating because just the DNA entered. But with a animal virus, the capsid makes it in. And so the virus, somebody, either a viral enzyme or a host enzyme, has to uncoat the viral DNA or RNA, has to get rid of that capsid. That's what uncoating means. Okay, getting rid of the capsid. Then, just like with phage DNA, biosynthesis starts, which means now the virus is going to take over the machinery of the host cell and start making more of its nucleic acid and start making more of the proteins that the virus needs for itself, whether that's capsid or other enzymes that it makes. Okay, so that's the same thing. So you can see I've sort of highlighted here in bold what things are different in animal viruses versus bacteriophage. Okay. Maturation just means all of those components get put together and assembled into mature viruses. And then release is a little bit different. Release is either by budding or by rupture. So rupture is kind of the same thing that bacteriophage do, where there's just so many new viruses that it just ruptures the host cell. But budding is a little bit different. Budding happens by envelope viruses, and it's kind of the opposite of fusion. So budding means that the new capsid, right, is going to approach the cell membrane. So here's the cell membrane. The capsid is going to approach, and it's just going to take some of that cell membrane with it. It's just going to bud off from the cell membrane, from the host cell membrane, and it's just going to take some of that host cell membrane with it, and that's what becomes the envelope of the virus. Okay, pretty cool. That's where the envelopes evolved from. Some virus sometime just said, hey, I'm going to take some of this envelope with me. It helps me get into the next cell, right? I didn't make that decision, but you understand what I mean by some random event that happened, and it became an advantage for the virus to have that envelope. Okay, so entry is different, uncoating is different, and budding is different from when compared to bacteriophage viruses. Okay, so let's take a look at some of these things. Okay, so here's an example of entry into the host. So here is the fusion. Okay, so you can't really see this here, but the viral envelope here is here, and it's actually just fusing with the host cell envelope, okay, or the host cell membrane, I should say. And by fusing, it just allows that the host cell envelope, the viral envelope just becomes part of the host cell membrane. And that means that the virus can just make its way in, because now the viral envelope has become part of the plasma membrane of the host cell. It just become part of that, and then that releases the capsid into the host cell, leaving the envelope behind as part of the host cell membrane. Get it? Okay, so that also leaves a space where the virus can then leave. Okay, but there's more to it than just that, because really one virus enters and maybe 1,000 viruses leave. So they all have to have a way to pick up some of that host cell membrane when they leave. Okay, receptor-mediated endocytosis just means that the capsid, the naked capsid, comes into contact with the host cell membrane and triggers that host cell to eat it, essentially, to bring it in by endocytosis. Okay, so it just has a way to trigger the host cell to do that by the way it binds to it. So the capsid will bind to the host cell, and then the host cell is by that attachment is going to open up and bring in that capsid by endocytosis. Okay, because there's nothing to fuse. There's no fusion that can happen with just a naked capsid because there's no envelope. Okay, so if you're confused, you want to see more, just look that up. Look up receptor-mediated endocytosis, or look up fusion of a viral envelope, and there'll be some videos, I'm sure, easy videos on YouTube that you guys can watch. Okay, so here's an example of budding. So that's how a lot of them enveloped viruses will escape. So here's a new viral capsid that has just been made inside the host cell. And then the virus will also, during the assembly and the maturation process, will put viral proteins, insert viral proteins, you guys, into the host cell membrane to tell the new viral particles where to exit. So it's going to exit where all these viral blue proteins are. So then when it comes to the surface, it's going to start to form this bud with these viral proteins in them, and then it will make this bud bigger, and eventually it will bleb out here to independently, no longer connected to the plasma membrane, but taking some of that plasma membrane with it, with those viral proteins or spikes now part of the envelope. Okay, cool. So many, many viruses would do this in one host cell. There would be these viral proteins, which would basically give the virus a visual of where it's going to exit the cell, because it's going to see these viral proteins in here. So it starts to push out. This is called a bud, and it's going to keep pushing out, keep pushing out, keep pushing out until it forms this larger bud and eventually releases. Okay, so here it is actually happening. This is lentivirus here. So here's the bud that's going to start. You can see the viral DNA here, and you can see the bud forming, and finally releasing. Okay, so look some of that stuff up, you guys. There's lots of, I'm sure there's lots of videos out there if you're confused or want to see it in motion. All right. Okay, so that's the main big picture of bacteriophage and animal viruses, right? The main steps, right, of attachment and then entry and then uncoding and assembly, maturation or biosynthesis, right? And then release, right? Those are the main steps of any viral infection. But we need to talk a little more about the biosynthesis step of animal viruses, because depending on what kind of nucleic acid they have, they replicate differently. So we're going to talk about DNA viruses. That's what's on the screen now. We're going to talk about RNA viruses, and RNA viruses have lots of different options. Lots of different options for RNA viruses. Okay, so we'll spend some time on those for the next several slides here. Okay. Okay, so DNA viruses are simple because they do the same thing that our genome does, because our genome is DNA, right? And so DNA viruses, once the DNA gets inside the host cell, they'll replicate their DNA inside the nucleus of the host cell using viral enzymes. So enzymes come along with, some enzymes come along with the virus, and they'll use some of their enzymes, they'll use some of our enzymes or whatever the host cell is. But because they're DNA, you guys, it's the same procedure as if our DNA was being processed. If it's going to be replicated, then there must be a DNA polymerase involved, right? If it's going to be transcribed, then there must be an RNA polymerase involved to make RNA, and the RNA will then need a ribosome to make the proteins. Okay, so it's the same stuff I've already taught you guys. It's just that it's the DNA virus that is taken over to do that. Okay, let's see, hang on a second here. Okay, sorry about that. Okay, so DNA viruses replicate their DNA inside the nucleus of the host cell using, it says viral enzymes, which is true, but it also will pirate and steal the activity of some host enzymes as well. Okay, my point here is though that because it's DNA, it's not that different from what our own DNA does, the host cell DNA does. Okay. Okay, so then once that DNA gets transcribed and translated, that will synthesize the capsid proteins in the cytoplasm using host cell enzymes. Okay, so viral enzymes inside the nucleus is typical, but host cell enzymes outside the nucleus is what makes the proteins. So host ribosomes, host tRNA, host RNA polymerase. Okay, so it uses a combination of both viral and host enzymes to make their parts. Okay, makes sense? But it's DNA polymerase here, it's RNA polymerase here, and ribosomes, right? Just like we've already learned. Just happens to be that the virus takes over the cell and makes its own stuff. Okay, so nothing too tricky about DNA viruses. Let me just show you a picture here. This is not from your book, but you get the idea here. So here's a virus here. It's a non-enveloped DNA virus. See, it's icosahedral shaped, right? Polyhedral shaped. It's going to attach to a host cell. Then it's going to enter the host cell and uncoat. So it's going to make its way into the host cell, and it's going to lose the capsid protein. See how the capsid is falling apart here? All these capsomeres in blue. And then there's the viral DNA. The viral DNA is going to make its way into the nucleus, all right? And it's going to start undergoing transcription, right? So there's the mRNA for the genes on the viral DNA. So it's going to go through transcription, and then that RNA is going to leave the nucleus to go out to the cytoplasm, and then it's going to be transcribed into new proteins. All right, makes sense? Just like in our cells. Some of the DNA, the viral DNA, then will also be replicated because it has to make new viral DNA. So you can see that's happening in the nucleus as well. So replication is happening, transcription is happening, translation is happening, okay? And then eventually maturation will happen where all the parts get assembled together, okay, to make new viruses. And sometimes that actually happens back inside the nucleus because the DNA can't get out. So you can see the capsid proteins make their way into the nucleus, these arrows are indicating here, and they start to surround or assemble around the viral DNA. And then once that happens, then the viral DNA will escape the nucleus, and then it will escape by release, in this case, from the cell to start again. Okay, so nothing too crazy, right? Nothing too crazy. The DNA makes it into the nucleus where transcription can happen, where replication can happen, just like any DNA would work. Make sense? Okay, so it's a typical DNA virus that attacks animal cells, and it's a typical way that these DNA viruses will replicate. Okay, attachment, entry, uncoding, biosynthesis of nucleic acids and proteins, maturation and release. Okay, so if we can block any of these steps, you guys, with a drug, then we can find an effective antiviral, right? That's what this is talking about here. So if we know how it replicates and we can block one of those steps, then we can stop the virus from replicating. Okay, so that's DNA viruses. It's the RNA viruses that are tricky, okay, or trickier. Okay, so I'm going to talk about 4 different types, really, kind of 4 different types here. So an unusual one are viruses that can multiply inside the nucleus of the host cell. So most RNA viruses, you guys, don't have to get inside the nucleus. Think about that. Why would an RNA virus, why would the RNA have to get inside the nucleus? Right? If it's already RNA, doesn't everything that happened to the RNA happen outside the nucleus? Right? Transcription or translation of the RNA happens outside the nucleus, right? Okay, so most of that stuff happens outside the nucleus, but some viruses like influenza virus is an RNA virus, okay, but it actually gets inside the nucleus to multiply. Okay, so that's a little bit unusual. Let me show you, I'll go back to this stuff in a second. Here's the influenza virus here. So it's one of the few RNA viruses that replicates in the nucleus of cells. So in an influenza infection, viral glycoproteins attach the virus to a host epithelial cell, like in our respiratory tract. That's how the flu initially attacks, right? So here it is attaching, okay? Because it's enveloped, it will be brought in by fusion, right? It's going to be brought in by fusion. It's engulfed and brought into the cell. So here it is, okay? The viral RNA and viral proteins are made and assembled into new virions that are released by budding. But the unique part, you guys, is that here's the virus, here's the RNA. It's going to come out of the virus. And instead of just staying out here to be translated, it actually will enter the nucleus. Okay, it enters the nucleus and then it gets replicated by RNA polymerase inside the nucleus. Okay. And then viral mRNA is used to make viral proteins. So 2 things happen, right? Either it's going to make more of itself by replication, or it's going to transcribe and translate its genes to make viral proteins, right? That's what the genomic nucleic acid is for. It's either going to be replicated or it's going to be expressed by transcription and translation. So both those things happen, but the RNA gets into the nucleus first, which is unusual. Okay, once that happens, right, then the RNA and the proteins will assemble into mature viruses and it will leave by budding, right? So new viral particles are made and released into the extracellular fluid here. The cell, which is not killed in the process, continues to make new virus. Okay, so that's interesting too. So it doesn't make enough that it kills the cell. It just enough gets made where it buds off and then the plasma membrane seals back up and the cell can make more viral particles. So that's pretty cool that the flu can do that, right? So it's not going to kill your cells. It's just going to use your cells to make more of itself. All right. So the unusual thing that you guys is that the mRNA makes its way into the nucleus. Got that? Okay, so let's go back a slide. And these are the other types of RNA viruses. Okay, so most of them then, not influenza or this group, but most of the rest multiply in the host cell cytoplasm. And they use an enzyme called RNA-dependent RNA polymerase. RNA-dependent RNA polymerase. That's unique to a virus. Okay, because normally in our cells, in other eukaryotic cells, you guys, what enzyme makes RNA from DNA? It's just RNA polymerase, right? So the RNA polymerase lands on the DNA at the promoter, and it can just start transcribing RNA, right? That's just called RNA polymerase. But if you start out as RNA, then you need a different polymerase to do it, right? Because regular old RNA polymerase needs DNA to sit down on and recognize. So you need a new RNA polymerase, and it's called RNA-dependent RNA polymerase, or RDRP. Okay, RDRP. Makes sense? So you need that enzyme to be able to replicate your RNA, okay, and make new RNA. Remember, the whole point of viruses, you guys, is just they're going to replicate themselves, okay? So all these guys use that, or at least, yeah, these two guys. We'll talk about this guy in a second. So here's another crazy thing about RNA viruses. Some of them are already in the proper, have the proper nucleotides, A's, U's, C's, and G's, right? The right code to be used directly as the mRNA for protein synthesis, right? So it's as if they were just transcribed from DNA, ready to go, like mRNA, ready to go to the ribosome and be translated. Okay, that's called sense strand RNA viruses. Those are called a sense strand or plus strand. So plus or sense strand means that they're in the configuration of the right code that they can be directly translated into protein. Okay, does that make sense? If it's in the opposite, if it's the opposite strand, right, the complementary strand, which happens, right, strangely enough, that happens, that means that it doesn't have the right code to be directly translated into protein. Instead, it has to be transcribed back into the plus or sense strand, and then that plus or sense strand serves as the mRNA for protein synthesis. Okay, so remember, like with the DNA, one strand is coded and one strand is template. So with RNA, if it's the plus strand, then that strand's good to go. It's already like the mRNA, it can just be directly translated by the protein, by that ribosomes. But if it's the opposite, if it's the complementary strand, that means it has the opposite nucleotides, right? So instead of having an A, it would have a U. Instead of having a C, it would have a G. So it's the wrong code. It's the opposite code. And so it has to first be transcribed to the plus or sense strand, and then that plus or sense strand gets translated into proteins. Okay, so let me show you a visual of that. That's this guy, figure 611. Okay, so here is your two options, right? So you have single-stranded plus strand RNA or single-stranded minus strand RNA. All right, so this is sense, this is antisense. This is RNA-dependent RNA polymerase, okay? And then here's translation. So if you're a plus strand, okay, if that's your type of RNA, then you can be directly translated into viral proteins, okay, because you have the right code. But if you're antisense or minus strand, you have to first be transcribed by RNA-dependent RNA polymerase into the plus or sense strand, and then that can be translated into proteins. Okay, does that make sense? Okay, you have to be this before you can be translated. This is key. You have to be plus or sense to be translated. You can't be antisense. It's the opposite. So you have to first go through transcription to become the sense strand, and then you can be translated. Okay, now why is this here though? Why is this arrow here? Because doesn't this guy have to make more of itself too? So think about what's happening in the cell. The virus has two jobs. It has to make proteins by translation, but it also has to make more of its genome. And to make more of its genome, that means it has to be replicated to this and then re-replicated to that. All right, so that's what's going to happen. So to make more of yourself, you have to replicate to the antisense, and then you have to re-replicate to the sense. All right, same thing with this guy. He has to make more of himself. So he's going to replicate to the sense and then re-replicate back to him for the antisense. Okay, so the difference is that before translation can happen, this guy has one extra step. Otherwise, they're the same, right? Otherwise, they're the same situation. They have to replicate themselves by doing this cycle. They have to make proteins by doing translation, but the antisense has to first be transcribed into the sense RNA before it can be correctly translated. Otherwise, it's going to have the opposite codons. It's not going to call for the right amino acid. It's not going to make the right protein, and the proteins won't be functional and the virus won't survive. Okay, so this is the extra step right here. This antisense DNA strand, RNA strand, has to first be transcribed into the sense strand, and then it can be translated into proteins. Okay, so let's see what it says down here. RNA viruses can contain sense single-stranded RNA that can be directly read by the ribosomes to synthesize viral proteins. That's this. Or they can be antisense and must first use the antisense RNA as a template for synthesis of the plus or sense strand before the proteins can be synthesized. Okay, so read that section in your book, you guys. That's on page, it's on pages, I don't even know. It's in section 6.2. All right, so if you have your textbook, it'll give you a little more information about that. Okay, this is tricky. This is the trickiest part of today's lecture. Okay, so I'll ask you on your test, what's the difference between sense and antisense RNA viruses and how they make proteins? I'll probably ask you that exact question on your test. Okay, so what is the difference between antisense and sense RNA viruses in how they make their viral proteins? Something like that. Okay, so let's go back. Okay, so here are the two types we just talked about. Sense means the viral RNA serves as the mRNA directly for protein synthesis. Antisense means that the viral RNA is transcribed first to a plus strand, and then that serves as the mRNA for protein synthesis. Okay, got it? This is the trickiest one right here. You can also have double-stranded RNA, you guys, but if you have both strands, then you have both of these and you're okay, right? You have the sense, you have the antisense. So you kind of work like double-stranded DNA, except you don't need to do that, except your RNA, but you still need RNA-dependent RNA polymerase to make more of your genome, okay? But you have both sense and antisense, and so they separate. RNA-dependent RNA polymerase will land on both strands and will start replicating in both directions and make more of each strand, okay? And then the sense strand serves as the template, or serves as the molecule to make protein, okay? It's this guy that's a little bit tricky, right? He needs the extra step. He has to first be transcribed to the plus strand before translation can occur. All right. Okay, and the last kind of RNA virus I want to talk to you about are called retroviruses. Okay, retroviruses. And these are RNA viruses that actually get converted to DNA first before they replicate, before they can do all the steps we've talked about. So we haven't talked about that category yet, right? We've talked about DNA viruses, and we've talked about RNA viruses that just stay RNA. But now we're going to talk about a category of RNA viruses that first will reverse transcribe to DNA and then use that DNA to make more of themselves. Okay, so they have a brand new enzyme that we haven't talked about yet called reverse transcriptase. So you have single-stranded RNA. It's not going to do any of the stuff we just talked about with sense and antisense stuff. Nope, it's going to instead reverse transcribe itself into DNA. So there's an enzyme that will make DNA from RNA. We haven't talked about this at all yet in any of our discussions, right? It's always DNA into RNA or RNA into RNA. Now we're talking about RNA getting transcribed to DNA. That's unique to these viruses, okay? It's pretty slick, okay? So they use the enzyme that they bring along with them to produce DNA from the viral genome, which is RNA, okay? Then that new DNA that gets reverse transcribed integrates into the host chromosome as a provirus. Remember with bacteriophage, we called it a prophage during the latent cycle, right? A prophage, when it integrates into the genome, well, it's a similar kind of thing, except we're not going to call it a prophage because we're not talking about bacteriophage. We're talking about animal viruses. So we're going to call it a provirus. Okay, does that make sense? So if it integrates into the host genome, it's called a provirus. And it may stay there, just like a prophage would. It may stay there for a while. Okay? So these are called retroviridae, that's the family name, or retroviruses for the common name. And HIV is probably the one you've heard of. Okay, so human immunodeficiency virus, it's the virus that causes AIDS or acquired immunodeficiency syndrome. Okay, and there are also viruses called oncoviruses that fall into this category too. Oncoviruses are viruses that cause cancer. Onco is that prefix that refers to cancer. Okay, so let me show you a picture of how HIV works. Okay, this is figure 612 in your textbook. Sorry, some of these figures, you guys, are horrible in the OpenStax book. But I think you'll get the idea here. Okay, so HIV is an enveloped virus. Okay, it's an enveloped RNA retrovirus. It's icosahedral shaped. You can see that capsid shape here. Okay, it's enveloped, and you can see that here. You can see these purple spikes. So these purple spikes, this one specifically is called GP120, which you don't have to know, will bind to a receptor on our T cells. T cells are part of our immune system, and this is the receptor here. And so this interaction is specific. So the GP120 will bind to the CD4 receptor here. All right, then it will eventually enter into the cell by fusion because it has an envelope. Okay, so here it is. All right, and then it will enter the cell. So here's the capsid. It will become uncoated. So you can see the RNA is being released from the capsid here. So here's the viral RNA. And these purple or black dots that you can hardly see are the reverse transcriptase enzymes that are already there, already made, already available for the virus to be able to start reverse transcribing its RNA into DNA. Okay, that's what's happening here. Okay, and eventually you'll get the viral DNA, double-stranded viral DNA here. All right, so that's the reverse transcriptase. So you have single-stranded RNA that's going to get reverse transcribed into double-stranded DNA. And then it will incorporate into the host double-stranded DNA here. It's made its way into the nucleus, and now it will incorporate and it will become a provirus. Okay, makes sense? So the viral DNA is transported across the nucleus and integrates into the host DNA. It's got another enzyme on it, you guys, called integrase, which allows that to happen. Okay. Then, just like any gene in the genome, the host genome, that the DNA from the HIV virus will get transcribed into new viral RNA, right? So here's the DNA. It's going to use the host machinery to get transcribed into new viral RNA. So it's making the same stuff it came in with, but it's using the host machinery to do that. Okay, so new viral RNA is used as genomic RNA and used to make viral proteins. So now you have the viral RNA that just got made from its DNA. That's crazy. And now it leaves the nucleus, and then it's going to translate that RNA to make viral proteins. And then assembly will happen, and maturation will happen. And sometimes that happens at the same time that budding is happening. right? So you don't have a mature virus yet, but it's already starting to bud. As long as it has all the components, you guys, it's good to go. And so eventually it will bud completely off and you have the mature virion, which has the envelope, it has the capsid, it has the RNA, okay? And some of the enzymes it needs to go infect a new cell, all right? But the key difference, you guys, is that the RNA gets reverse transcribed by the existing enzyme called reverse transcriptase that the virus carries along with it to make double-stranded DNA to get incorporated into the host cell. The host cell then as transcription happens in the host cell, it will make new viral RNA. That RNA will then make new proteins, and all of it will get assembled into new viral particles. Okay, that's pretty nifty, pretty unique kind of mechanism there. Okay, so 4 different things we talked about, you guys, for RNA viruses. Just to recap, right? So we have a few that will multiply inside the nucleus of the host cell, okay? And then, but most are in the cytoplasm. So then you have this guy, or these 2, which are single-stranded RNAs. And one of them is a sense strand, one of them an antisense. This guy can be directly translated. This guy has to be transcribed first into the sense strand, and then it can be translated. This guy has both strands already, so he's good to go, right? So actually that's kind of 5, right? 1, 2, 3, 4, and then we have this guy, the retroviruses. Okay, so lots of different ways that RNA viruses can go through biosynthesis. Okay, so spend some time on that. Look up some videos. If you find good ones, send them to me and I'll post them. Okay. All right, so the next few slides, you guys, I just wanted to show you some examples of viruses and just show you how they're categorized, right? So you should know what everything on the left side means. Single-stranded DNA non-enveloped, right? There's an example. Double-stranded DNA non-enveloped. Double-stranded DNA enveloped, okay? And then here's some examples and some important genera or genuses. The plural of genus is genera. You don't have to know this. I just want you to see all, how all the different viruses are categorized, and you can see their various sizes, all of them in the nanometer ranges, right? Really small. Here's single-stranded RNA plus strand non-enveloped, single-stranded RNA plus strand enveloped, right? Antisense, 1 strand of RNA. Antisense, multiple strands of RNA. Single-stranded RNA produce DNA, these are the retroviruses, or double-stranded RNA non-enveloped. Okay, crazy, right? All kinds of categories, you guys, of different viruses. And you can look through here and see where your favorite virus falls. Okay, let's see if I can see coronavirus in here. It's an RNA virus. Here we go. Okay, so the coronavirus, which is the family that one of them causes COVID-19, falls into the single-stranded plus strand enveloped virus category. Okay, so you can see SARS here. That's a type of coronavirus. And if this was more up to date, we'd have COVID-19 in here as well. Okay. Okay. Okay, so that was the hardest part of the lecture. I just have a few more things I want to talk to you guys about before we finish for today. Okay, so the next topic I want to explain are viral infections that can be called latent and some called persistent. So we have persistent and latent viral infections, okay? So latent just means that the virus remains in an asymptomatic host cell for a long period of time. So kind of like dormant, right? And they may stay like that forever. You may never feel the effects of the virus because it may never replicate. But if something changes in your body or in the host cell, then it may reactivate and start to replicate again. Okay, and then you'd feel the symptoms, and you would have some kind of negative effect most likely from that virus, and you'd be infectious again most likely. So cold sores and shingles are 2 diseases that are caused by viruses that are latent. So if you have cold sores, that the cold sores are caused by herpes simplex virus. So that can affect typical regions are either genital herpes or oral herpes. Okay, so they're also called cold sores if they're on your mouth, and they recur. So if you have this, it's very hard to get rid of. And under times of stress, that you may get a cold sore. So it could be when you're stressed. It could be when you are have a lot of UV exposure, right? People can often feel when they're going to get one because it starts to tingle. Okay, so that virus that causes that herpes simplex virus, is latent in those cells. Sorry, and it gets reactivated under times of stress. Shingles is just a reactivation of the chickenpox virus. So if you had chickenpox as a kid, many of us did. You might not have, because the vaccine became available probably when most of you were kids, or many of you. But I didn't. I had chickenpox, which means I have that virus in me, and it could reactivate as shingles. So I just recently did get my first shingles vaccine at age 50. That's when you're supposed to start getting that. So I don't want shingles. It's much more severe and painful and debilitating than chickenpox. So I definitely don't want that. Okay, but that's what a latent virus means. You may stay asymptomatic for weeks, for months, for years, or forever, right? So we all have viruses in us, you guys, that are latent, that we may never know again. Okay, as opposed to a persistent viral infection, which just means you always have the symptoms and they get worse and worse and worse. And if there's no treatment, then it's generally fatal. Okay, so if there's no treatment, or you can't be treated, or the treatment doesn't work, then eventually it will become fatal. So it's just a persistent viral infection that gets worse and worse over time. So that could be months, it could be years, but it's just called a persistent infection. Okay, so this graph shows a sort of, a simple way to view that, right? So when you first get an infection with a virus, you guys, it's called an acute infection. Okay, so the number of virus particles is very high when you first get infected with a virus, typically. Okay, so for most viruses, when we're cured, they go away and we don't have them anymore. But if it's a persistent infection, then it may continue and it gets worse and worse and worse and worse. So eventually your body fails and the virus particles increase, and it can cause death unless there's a treatment. Okay, if it's a latent infection, it means you get an initial infection and it goes away, but it hangs out, but you don't know it. There's no symptoms until it recurs for some reason, and then you do get a latent infection. So you can see the virus particles go up. So if your initial cold sore, you might get like 4, right? And then period of time goes by, months, years, days, whatever, and then maybe you get 1, right? And so you don't have quite the same infection or the same number of viral particles, but you definitely see an effect. Okay, hang on one second. Okay, so does that make sense for persistent versus latent? All right, so persistent is usually it's a one-way street, right? It's not just going to go and come back and be nothing and then come back. It's something that just persistent. It's just persistent in its gradual sort of getting worse, and that's what this blue line kind of shows you here. Okay, whereas latent, you don't have any symptoms maybe for years, and then you might have symptoms, and then it goes away, and then you might have symptoms again, and then you might have symptoms again. Okay, so here's an example. The one I was talking about on the left here is the virus that causes chickenpox. It's called varicella zoster. So it's an enveloped icosahedral capsid virus. So you should know what that all means now, right? So you can see it here. So here's the capsid, here's the envelope. Okay, it's double-stranded DNA, and it becomes incorporated into the host DNA, making it a provirus, right? And that's how it stays latent. It stays in the DNA until something might trigger it, a stress, and then it pops out of the host genome and starts to replicate, and that's when you'll get symptoms. That's when you get symptoms again, right? So this is over here shows you what happens when it does pop out of latency, and you can get these blisters, and this is shingles. Okay, so shingles is a reactivation of a latent form of the chickenpox virus. So it's the same virus, but it reemerges as something different, right? It's a different rash. It's painful. It's usually on one side of the body. It can be debilitating. It can be painful just to even put a shirt on over the rash. Okay, whereas chickenpox are usually just itchy for like a week. All right, so it's a different kind of it manifests differently when it comes out of latency and reactivates. Some viruses come back the same, but this one happens to come back with very different symptoms. Okay, so there is a vaccine available now. It's a 2-shot series that you get 2 to 6 months apart, and they recommend it when you turn 50 because that's the most common age when you might start to get see shingles reactivating. Okay. All right, so here's some examples of latent and persistent infections, you guys. So we talked about cold sores and shingles. Leukemia can also be a latent infection caused by this virus here, HTLV1 and 2. Don't have to know this, but the symptom there is increased white blood cell growth. Okay, here are persistent diseases. Okay, and then here's the virus that causes them. And here's what happens in that persistent sort of disease state, what happens, right? So cervical cancer, which is caused by human papillomavirus or HPV, increased cell growth causing tumors wherever that virus has infected. Again, it can lead to like cervical cancer, okay? HIV, decreased T cell count, right? And that's caused by HIV 1 and 2. Liver cancer can be caused by hepatitis B. Persistent enterovirus infection can be caused by echoviruses. Okay, so just some examples of persistent and latent viruses. Okay. Okay, I don't have a heading here. Okay, well, the heading of this should say viruses and cancer, because that's kind of where I was leading here. So if you look back here, let's see. Okay, so some of these here, viruses cause cancer. So that's where it's leading me into the next part of my talk here. So I'm not sure why. There we go. Came out weird. Okay, so viruses and cancer. So there's several types of cancer, you guys, that are caused by viruses. So remember, we've talked a little bit about cancer before. Cancer is caused by mutations. So those mutations I talked about in, let's see, it was in lecture 7, probably 7, part 2. It can either be caused by spontaneous changes to our DNA or some kind of mutagen. And sometimes that mutagen, you guys, is a virus. So if that virus recombines into our DNA, in a gene, an important gene that is involved in suppressing cancer from happening, then it could lead to cancer, right? Because if you mess up with that gene by inserting into it, then that gene no longer is going to work and cancer could occur. Does that make sense? So it's always viruses that will recombine into our DNA, the provirus. Okay. So there are several types of viruses, of cancer that are caused by viruses, and it can develop long after the viral infection has occurred. You know, so if you get an HPV infection and you have genital warts, those symptoms may be long gone, but it could be years after that you may end up with tumors growing, right? Cancer growing. So it could take a while for that to happen. Okay, so cancers themselves are caused by these viruses are not contagious. Now, the virus can be contagious, right? You can pass the virus, but the cancer that's caused by that virus is not contagious. Make sense? Okay. So if you're a virus that can lead to cancer, you are called an oncogenic virus. Oncogenic. Onco means cancer. So oncogenic means to cause cancer. So if you're a DNA virus that can recombine into our DNA and cause cancer, then you're called oncogenic. And so here's some examples. Again, you don't need to memorize any of these, you guys. It's just for your information. Okay, these are some common oncogenic viruses. Viruses that when they incorporate into our DNA can lead to uncontrolled cell division. That's what cancer is. Tumors are just uncontrolled cells. So normally we have lots of checks and balances of when our cells divide. In cancer, we don't. So if you're an oncogenic virus, you are incorporating into a gene that is involved in controlling cell growth. So if you incorporate into that gene and recombine into that gene, you've just messed up that gene. No longer is cell growth controlled in that of that cell because of that virus. And so now the cells can grow out of control and tumors can result. Okay. So how does it happen? Okay, so the transformation of normal cells into tumor cells is how cancer happens. So as I already said down here, if you're a virus that becomes integrated into the host cell's DNA to induce tumors, you're an oncogenic virus. Okay, so that can trigger what are called oncogenes in the host. So this is the name for the virus. If you're a gene in the host that's important to preventing cancer, you're an oncogene. Okay, an oncogene is a host gene that's important in preventing cancer. But if an oncogenic virus incorporates into you, then you don't have control anymore. And then you can start, these cells will start transforming into cancer cells. Does that make sense? So oncogenes are normal host genes, you guys. They're normally there doing things that prevent cells from growing out of control. They are the policemen of the cell cycle to make sure that we're not reproducing when we're supposed to be, or we're not supposed to be. Make sense? So when everything's normal, these genes act as sort of stop signs to say, hey, we don't need you to replicate. We don't need you to make new cells, stop. But if a virus, an oncogenic virus, incorporates into that gene, into that oncogene, now it doesn't work anymore. And so that gene can no longer produce the protein to stop cell division. And so cells start to grow out of control. That's a tumor, okay, which can lead to cancer if that tumor spreads. Okay, so how does that happen? Oncogenes can change by one of three ways: spontaneously, right, a mistake in DNA replication, by some environmental mutagen like UV light or cigarette smoke, or those other things we talked about when we talked about mutations, or the new one that we've talked about today is viral infection by one of these oncogenic viruses. Okay, so by incorporating into these genes, you've messed up that gene. You can't produce the protein that acts as the policeman to stop cell division. Okay, that's how cancer happens. Make sense? Okay. Oh yeah, okay, so that's it for viruses. Okay, so my last few slides, you guys, I'm going to talk about a few other acellular pathogens that aren't viruses. They're just other weird things that exist in our world, okay, but they cause lots of damage. Okay, so we didn't really talk too much about plant viruses, you guys. So we're going to talk about plant viruses and viroids and something else called virusoids. Okay, virusoids. So that should be on this list too, but I'll show you at the bottom here. So all of it's on one slide because I'm not going to spend too much time on this. Okay, so plant viruses. Plants are hard to infect with a virus because they have a really, really thick, waxy outer coating and a really thick cell wall, right? So they have cellulose in their cell wall. They have a waxy cuticle outside of that. So it's really hard for viruses to penetrate into those cells. They're really strongly protected. So there has to usually be some kind of wound to the plant, or insects will penetrate, with a stinger or some kind of proboscis or some way that they can inject a virus into the plant. Okay, so that's really all I want to say about plant viruses, but they exist. We know they exist. Farmers deal with this all the time with their crops. Okay, but in general, they're pretty well protected, so they don't have nearly as many problems as do animal cells, because animal cells are not very hardy, right? We don't have a cell wall. We don't have a waxy coating. Okay. Okay, there's these other things called viroids. Okay, so not plant viruses. They're just short pieces of naked circular RNA. So no capsid around the virus, no envelope around the virus, just naked circular DNA or RNA. All right. They can self-replicate. They don't have a protein coat. As I said, they're naked, right? But there's lots of them, and they can result in devastating losses to agriculture. Okay, so farmers are dealing with this a lot. They can cause lots of damage and spread really quickly in certain crops. And so, farmers or any agricultural company is typically worried about these kinds of things, and it's hard to control them. Okay, it usually wipes out a whole crop before it's done. Okay, so those are viroids, and similar to that are virusoids. which are kind of the same thing, except they can't self-replicate. All right, they cannot self-replicate. They're single-stranded, but they need help to replicate. These guys can replicate on their own, but virusoids need a second or helper virus to replicate. All right, so they need a second virus to co-infect the plant with this virusoid to be able to replicate. So it's probably lacking some kind of enzyme to replicate on its own. So it needs a helper virus to do that. Okay, so that's the difference. It's not very common, but the ones that are out there are also equally can be equally damaging to crops. Okay, so viroids and virusoids are similar. They're RNA, but virusoids need a helper virus to replicate. Okay, and plant viruses, remember, if you're a virus, you have to have at least DNA or RNA and a capsid, right? Maybe an envelope, but at least a capsid. So these guys have capsids. These two are naked, no capsid. All right, makes sense? All right, but they're all kind of affect plants. Okay, and the last thing I want to talk about, which is also part of your homework, you guys. So there's like a little quiz that's in this module that deals with prions. Okay, so it's also in your textbook, so you can look there too. I'll just explain it to you guys, and then you should be able to easily take this quiz. Okay, so prions are just proteins, no nucleic acid. So this is the first one we've talked about that's like that, right? So no nucleic acid, just protein, and they cause disease, which is really wacky, right? So you have things that are just RNA. You have things that are just protein that are causing disease, which is pretty incredible, I have to say, right? That cause these problems. Okay, so what are prions? They're proteinaceous, protein, infectious particles. And the prion comes from P-R-O and I-N, although it would be prone then, right? So I'm not sure what happened there. There's probably a story behind that, but they're called prions. All right, so what are they? Again, they are just malformed proteins. So there's a normal protein that exists in our brain tissue, in our spinal cord. But a prion is just one of those proteins that is shaped incorrectly. But then it can induce neighboring normal proteins to also form incorrectly. And that can cause disease and death. Okay, so how do we get them? So they're either inherited. So we might inherit this from a parent, and they're transmissible. So they can be what's the word I'm looking for? They can be shared either by ingestion. So you're eating something that contains prions, a transplant. So if you get a transplant with some kind of tissue that contains these proteins, you can get them that way. By surgical instruments. So if the surgical instruments have prions on them, you can get infected with them if you undergo a surgical procedure. So they cause something called spongiform encephalopathies. Spongiform encephalopathy, that's a mouthful. So encephalopathy just means disease of the brain. Okay, so apathy refers to disease, and encephalo refers to the brain, and spongiform refers to what it makes it look like. It makes holes in your brain tissue like a sponge. So it's called spongiform encephalopathy. Okay, here's some common names for them. You've probably heard of mad cow disease. This spread many years ago now by infected cattle or infected beef that was not processed correctly, and some of the brown beef got some of the brain tissue got mashed into some of the ground beef, and so that spread, especially in Britain. There's Creutzfeldt-Jakob's disease. There's Gerstmann-Straussler-Scheinker syndrome. Okay, these are all human diseases. Sheep scrapie is typically in sheep, but it can be passed to humans too. Fatal familial insomnia. So these are all just different types of prions, but they all cause pretty much the same outcome of holes in your brain and death. There's no treatment. Okay, so this is not a curable disease. It's pretty awful to go through it. Okay, so what happens? So here's the normal protein, you guys. It's called PRPC. PRPC, normal cellular prion protein. Okay, normal cellular prion protein on the cell surface of brain tissue. Okay, so that protein is on the cell surface. Okay, so it can mutate though to a rogue protein Okay, a second protein called PRPSC, which means scrapie. So scrapie protein. So this SC subscript or superscript here refers to scrapie. Okay, so that just means that this normal protein mutates to this rogue protein that's no longer functional, but also causes the spread of this protein, and it results in killing the brain tissue in that area and creating sort of holes or a sponge-like look to your brain tissue. All right, so this accumulates in your brain cells and it forms plaques. Just like we talked about plaques on those bacteriophage plates, same idea, sort of holes in the tissue there. Okay, so it creates a sort of spongy looking brain tissue. It's infectious, which means it can be passed to the next normal protein and the next normal protein. So one scrapie protein can induce the normal neighbor protein to become a scrapie protein. So it's infectious in that way, which leads to more plaques. Okay, so it can spread pretty quickly to all the normal cellular prion proteins in your brain. Okay, so it causes, like I said, transmissible spongiform encephalopathy, TSE, because it can be passed from one protein to the next. It kills brain cells and forms holes in tissues. It leads to, at the very least, brain damage, but often loss of motor control and dementia, and eventually death. There's no cure. Okay, so it's pretty sad to watch people suffer through this. It doesn't take long, but it can be months, maybe years, but usually months to watch people deteriorate with this disease. One of the worst stories I've heard is how it was passed. Is there was tradition in some, I don't know if it was in New Guinea or someplace in Africa. I get my stories mixed up, but one of the traditions when you buried your loved ones, one of the traditions was to eat their brain as to honor them. And this person happened to die from a prion-related disease, from a TSE, spongiform encephalopathy. And so everybody in the family who ate part of their brain also got it and also died from it. So that's another way it can be passed. So not a great way to go, especially when your whole family gets it. So let's just take a look at a really simple figure here to explain it. Here's what it looks like. So normal brain tissue, you guys, should be all pink. It should be all pink in here, but you can see these white plaques, which is where the scrapie protein has destroyed the brain tissue there. Okay, so here's your endogenous normal prion protein here in green, right? But then it may come in contact with this scrapie protein, right? And so this scrapie protein can be either from a spontaneous mutation that occurs in the normal protein, right? Or there can be a conversion of mutant PRP into this protein, or you can get it by ingesting it. So inoculation of this protein. So there's several ways that this can happen. But however it happens, once it comes into contact with the normal protein, it will convert that normal protein to the scrapie or the rogue protein here. And then those guys will replicate, and they'll keep doing it and keep doing it and keep doing it as the normal proteins come into contact with the scrapie proteins. And then you get this accumulation of these scrapie prions, and that's how you get these plaques. Okay, so eventually that will lead to all kinds of issues when you start losing brain tissue. Okay, so down here you can see here, the scrapie protein may arise spontaneously in brain tissue, especially if a mutant form of the protein is present, or it may originate from misfolded prions consumed in food that eventually find their way into the brain. That's how the mad cow disease outbreak happened, from eating ground beef that had brain tissue in it that was contaminated with these scrapie proteins. Okay, so again, no RNA, no DNA in these things, but look at the disease, look at the trouble they cause, right? So no genetic material at all, just this rogue mutant protein that can convert normal proteins to itself, right? So there's a good mechanism there that I'm not talking about how it does that, but that's a pretty unique pathogen that we haven't really seen before. Okay, that's the end of lecture 10, you guys, and also the last lecture before your second exam. Okay, so I will see you guys next time.